They called her a queen who could charm an empire. They also told stories about the blood that made her family into something other than ordinary rulers. Whispers that the dynasty's children looked strange, even eerie, as if the house had paid for its power with a hidden price. That is the claim behind the bold question in our title, the genetic revenge, the terrifying reason Cleopatra's children never looked human. It is a question built from three kinds of things: images, rumors, and the cold arithmetic of heredity. Today, we will take each of them apart. Imagine a Mediterranean throne room under a sun so bright it makes the gold of the floor flash like water. The Ptolemaic court in Alexandria was a spectacle of borrowed gods, Macedonian blood, Egyptian rites, and Greek language, a hybrid kingdom born from Alexander's generals and rooted in a land older than their bloodlines. In that hybrid world, appearance was not a private matter. A royal face was a political instrument, minted on coins, carved into statues, shown to allies and enemies alike. To rule meant to be seen as legitimate, and that visibility made the bodies of the royal family into evidence, propaganda, and rumor fodder all at once. Cleopatra VII sits at the center of this story. Daughter of a dynasty that married within itself to keep power consolidated, she lived at the intersection of Hellenistic kingship and ancient Egyptian theocracy. Her life is relatively well documented for an ancient queen: her alliances with Julius Caesar and Mark Antony, her political maneuvering, and the children she bore. A son known historically as Ptolemy XV Philopator Philomator Caesar, popularly called Caesarion, and 3 children fathered by Mark Antony, usually given the names Alexander Helios, Cleopatra Selene, and Ptolemy Philadelphus. Those children, like their mother, were objects of intense public scrutiny, but what can the sources and science truly tell us about what they looked like? First, the sources. Ancient authors wrote about Cleopatra and her family through lenses that were never neutral. Roman historians and moralists treated her as exotic, dangerous, and often morally culpable for Rome's troubles. Plutarch, writing in the 1st and 2nd centuries after the events, gives us portraiture of personality and politics more than clinical descriptions of physiognomy. Cassius Dio and others relay rumors in Roman propaganda that amplified whatever could serve Rome's narrative of foreign peril. In many of these accounts, Cleopatra's sexuality, her theatrical persona, and the spectacle she staged around her children receive as much attention as their bodies. Sometimes the rhetoric slides toward descriptions of oddity or deformity, but those descriptions are political currency. They tell us less about inherited traits than they tell us about how Rome wanted to depict a powerful foreign queen and her heirs. When we switch from text to image, the trail becomes both promising and treacherous. The Ptolemaic dynasty left coins, busts, reliefs, and sculptural portraits that survive in museums and storerooms. Coins are especially important because they carried the ruler's face across the empire. A portrait on a denarius or tetradrachm was an official image meant to repeat and normalize, to remind subjects of who ruled. But portraiture is not neutral transcription. The same hand that carved a triumphant, ingeniously coiffed Cleopatra into silver might deliberately flatten, idealize, stylize, or emphasize features to communicate political signals. In the Hellenistic world, the conventions of portraiture included aging a ruler into gravitas, or smoothing features to suggest divinity. Sometimes children were depicted with disproportionately adult features, or were represented symbolically rather than realistically. So when a modern viewer sees an odd face on a coin and wonders if it reflects a genetic abnormality, we must pause and ask, is this artistry, iconography, or anatomy? Now consider the dynasty's marriage practices. From the earliest Ptolemies, sibling and close kin unions became routine. These marriages were political, tools to concentrate legitimacy and property. They were not unique to the Ptolemies. Egyptian pharaonic tradition included sibling unions, and neighboring dynasties engaged in consanguineous marriage as well. But what does consanguinity mean biologically? How does it translate into the visible features of a child, much less a line of children across generations? Genetics gives us a vocabulary to describe risk without turning it into scandal. When close relatives reproduce, the chance that they both carry the same recessive gene increases. A recessive mutation usually hides in a carrier, causing no trait unless an individual inherits 2 copies. If both parents carry the same recessive variant, each child has a roughly 25% chance of expressing the trait. Multiply that risk across generations and across many shared ancestors, and the probability of homozygosity for rare variants climbs. Long-term inbreeding increases homozygosity more broadly, which can raise the risk of congenital disorders, reduced fertility, developmental problems, or morphological traits that deviate from familial norms.
But risk is not inevitability. Genetics deals in probabilities, not moral judgment or dramatic certainties. It is tempting to line up the dynasty's documented sibling marriages and draw a straight arrow to physical degeneration. 6 generations of close kinship, the argument goes, could concentrate deleterious variants and produce children who looked odd enough to generate rumor. That is plausible as a mechanism, but to transform plausibility into proof requires data the historical record mostly does not provide.
There is a critical absence here. Securely identified skeletal remains from the Ptolemaic royal line, preserved in contexts that allow confident attribution, are not available from modern genetic testing. Without bone or tooth samples from known Ptolemies, there is no direct genetic evidence to sequence and test. Ancient DNA, or aDNA work in Egypt, has been improving. Recent studies have successfully sequenced genomes of some ancient Nile Valley populations. But royal burials, especially from late Hellenistic Alexandria, present unique obstacles. Tombs were looted, the climate accelerates DNA decay, and chains of custody for artifacts can be murky. To make the claim that Cleopatra's children bore visible genetic disorders would require, at minimum, remains that archaeologists can securely connect to a named royal, successful aDNA retrieval from those remains, and an interdisciplinary comparison between genetic findings and osteological markers. Until those elements exist, any genetic claim about Cleopatra's line remains speculative, possible, maybe probable in narrow scenarios, but not proven. If we cannot yet test royal bones, what other evidence can we bring to bear? The answer is the triangulation of 3 imperfect sources: texts, images, and comparative historical examples. Each brings something of value and something of risk. From the textual side, the ancient writers give us a palette of narratives. Plutarch paints Cleopatra as charismatic, cunning, and theatrical, a queen who staged her image with intent. Cassius Dio, whose account blends fact and rumor, reports Roman disgust and astonishment in equal measure. Even when these authors describe physical traits, their perspective is embedded in context. They are telling stories to explain political outcomes, not to compose medical case histories. That means their observations must be read as rhetoric as much as reportage. Quotes or paraphrases used as evidence must be balanced by skepticism about motive. When a Roman historian emphasizes the foreignness of Cleopatra's children, that emphasis can be propaganda aimed at discrediting her and her heirs. It is also the source of much of the modern fascination. Ancient slander tends to linger, passing into the long shadow of history. From the visual side, coins and portraiture are archives of statecraft. A tetradrachm bearing the queen's profile circulated through marketplaces, temples, and foreign courts. But portrait types in the Hellenistic world often followed conventions: stylized hair, repeated facial angles, and symbolic markers that had more to do with dynasty than with fidelity to an individual's physiognomy. In some cases, younger rulers were given adult traits to convey authority. In others, stylization made faces remarkably similar across generations, a house style that emphasized continuity over accuracy.
Statues and busts go through periods of restoration and reinterpretation. Many of the pieces we see today have been repaired, reworked, or even misattributed. That complicates any direct read of features as genetic evidence. Then there are comparative historical examples, where inbreeding within royal houses is better documented, and in some cases better studied. The Habsburg jaw in early modern Europe provides a cautionary model. Long-term dynastic intermarriage did coincide with a recognizable phenotypic cluster: mandibular prognathism, dental malocclusion, and sometimes fertility problems. Modern genetic work has connected some of these traits to inherited patterns. But the Habsburgs are a distinct case. They lived in a cold, well-documented era, with abundant portraiture and better preserved remains. Using them as a direct analog risks overstretching the evidence. The Ptolemaic world is different in epidemiology, environment, and funerary fate. Analogies illuminate possibilities, but cannot substitute for direct proof. So far, this is a forensic inventory. Here is what we have, here is what we lack, here is what might connect the dots. But the story that makes the title dramatic is not solely about biology. It is about image production and political language. A Roman writer could make the offspring of a foreign queen seem grotesque by comparison to ideal Roman norms. A coin could flatten a child's features into a symbol. A rival court could plant whispers to delegitimize heirs. Political enemies weaponized appearance, the way modern campaigns weaponize imagery. To understand whether there was a genetic revenge, we must read the politics of the portrait alongside the mathematics of inheritance. Consider a specific example of visual ambiguity. Coins struck in Alexandria during the late Ptolemaic period sometimes show youthful male profiles with heavy jaws or pronounced brows. A 21st century eye might look at such reliefs and feel a chill. These faces seem oddly proportioned when compared to canonical Greek ideals. But here is the crucial question. Were those proportions an artist's shorthand, a deliberate mark of stoicism, an attempt to fuse Macedonian lineage with Egyptian iconography, or evidence of a familial trait? Art historians warn about treating portraiture as a mirror. They remind us that technical constraints of die engraving, regional stylistic traditions, and the propagandistic need to signal continuity all shape portrait types. The same die could be used for multiple issues over decades, preserving a stylized face across generations. When the same facial formula repeats, it may say more about the dynasty's iconography than about DNA. And yet, the repetition of a face across coins can have an effect on perception. If many portraits from a dynasty carry the same unusual features, observers across centuries can begin to conflate style with anatomy. Rumor finds fertile ground here.
Roman satirists and moralizers, looking for a story that explains Rome's entanglement with the East, could then add a layer of sensational commentary. Children who did not look right were taken as evidence of moral corruption, divine punishment, or exotic otherness. The rhetoric amplifies the image confirms the rhetoric. The cycle becomes self-sustaining. This is why an investigative approach must bring modern genetics into the room, not to close the argument with a neat diagnosis, but to lay out the mechanics and limits of what biology can tell us. Genetics offers language, heterozygosity, homozygosity, recessive alleles, inbreeding coefficient, founder effects. It also offers tools, whole genome sequencing from ancient remains, analysis of runs of homozygosity to detect long-term consanguinity, and the comparison of allele frequencies across populations to infer ancestry. But each tool comes with caveats. Ancient DNA degrades, contamination from modern handlers is a persistent hazard, and even when sequences are retrieved, linking particular genotypes to specific phenotypes, especially complex facial features, is fraught. Many traits are polygenic. They arise from many genes interacting with environment. A protruding jaw could be partly genetic, partly the result of early childhood disease, nutrition, or dental issues. Facial morphology is a complex outcome, not a simple genotype to appearance line. To turn back to the Ptolemaic children, what would definitive evidence look like? First, archaeologists would need to identify remains that can be convincingly tied to named royals, burial context, inscriptions, or associated grave goods that leave little room for doubt. Second, those remains would need to yield retrievable DNA that is demonstrably endogenous and not modern contamination. This requires careful excavation, proper handling, and modern laboratory protocols. Third, geneticists would have to find either known pathogenic variants linked to recognizable syndromes, or a pattern of genome-wide homozygosity consistent with generations of close-kin marriage. Fourth, osteological evidence would ideally match genetic findings, cranial facial anomalies visible on bone, dental pathologies, or developmental markers in the skeleton. Only the convergence of these lines, securely attributed bones, clean aDNA sequences, congruent osteology, would allow a confident statement that a hereditary disorder produced a visible, repeated phenotype among the Ptolemaic children. None of these convergent conditions is currently met for Cleopatra's immediate offspring. That absence should temper our hunger for a sensational verdict, yet the absence of proof does not mean the hypothesis is impossible. Consanguinity creates biological risk. The Ptolemaic marriage practices created social conditions that could raise that risk. The ancient record contains rhetoric that sometimes implies deviation from contemporary norms. Coin portraits sometimes look unusual to modern eyes. Those three threads, social, textual, and visual, are enough to make the question worth investigating with rigor. They are not enough to deliver a final answer. What follows, then, is a deeper look into the evidence itself. We will move through the visual dossier, coins, busts, and the artistic conventions that shaped them, and then into a compact genetics primer that explains the essential mechanics in plain terms. Along the way, we will bring in voices from historians and scientists who have studied Hellenistic portraiture, consanguinity in royal houses, and the challenges of aDNA in Egypt. Where the sources are absent or ambiguous, we will be explicit, indefinite. Where the odds are mathematical, we will say so. But before we dive into the visual archive, pause for a moment and consider what is at stake in this inquiry. This question, did Cleopatra's children look different because of the dynasty's inbreeding, is a test case for how we treat bodies in the past. When we imagine a child's face as either a scientific fact or a moral indictment, we are deciding whether to let curiosity become spectacle. We are deciding what counts as evidence and how much power we give to rumor. The temptation to fill gaps with dramatic narratives is strong, and the past, especially a past saturated with politics, often rewards the dramatic story with attention. That reward, in turn, encourages more dramatic stories, and the loop tightens. If this investigation has a moral as well as a methodological aim, it is this: to separate reasonable inference from alluring fiction, and to respect the humanity of the individuals who populate our sources, even as we interrogate the mechanisms that shaped their public faces. Cleopatra's children were heirs, not specimens. They were imagined by enemies and praised by allies. Their image was a political tool and a private bond. The point is not to sanitize history of its uglier patterns, but to resist turning those patterns into a macabre parable about biological determinism. If you like investigative history, subscribe for more deep dives that question sensational claims and follow the evidence where it leads. We begin our close reading of images with coins, the most common visual documents that survived from the Ptolemaic court. Coins are not portraits for pleasure, they are statements of authority. A die cutter in a mint had to work in metal, with tools that flattened features into relief. The practical constraints of minting introduce stylistic simplifications. Noses become strong lines, eyes become apertures, hair is rendered as patterned locks. In the Ptolemaic case, there is also an overlay of Egyptian regal symbolism. The portrait of a Ptolemaic sovereign could be made to echo pharaonic conventions, a frontal gaze here, a stylized wig there, in order to speak to Egyptian subjects.
That hybridity can produce faces that look at once Greek and Egyptian, neither of which maps directly onto modern notions of realistic portraiture. Examine, for example, a series of tetradrachms purportedly issued in Alexandria in the late 1st century BCE. Side by side, the male profiles show a repeated formula: a prominent brow, a squared jaw, and a mouth set in a firm line. To a modern viewer steeped in classical ideals of facial proportion, those features may seem exaggerated, but art historians point out several technical factors. Coin dies could be reused across decades. A deeply carved die throws a severe shadow that exaggerates relief. Regional workshops favored certain facial templates, much like a photographer today might apply a signature filter. The repetition may therefore reflect die reuse and aesthetic norms rather than familial pathology. Moreover, the numismatic record is incomplete. We only see a fraction of what was struck, often surviving through chance or hoarding. Selective survival can skew perception. Beyond coins, surviving sculptural fragments and busts present a more complicated picture. Marble and bronze offer sculptures greater latitude to model faces with subtler three-dimensional cues, but they also invite restoration. Many ancient sculptures were repaired in later centuries, parts replaced or reshaped to fit contemporary tastes or collection needs. A head that appears oddly proportioned today may have lost a nose, had a forehead reworked, or been modified to mimic a more fashionable type in Rome. Attribution is also a problem. Objects labeled Ptolemaic may come from workshops influenced by the dynasty rather than from royal commissions. The result is a visual landscape that is noisy and uncertain. This is why image evidence must be read in context. Coin die series with documented provenance, sculptures excavated in secure archaeological contexts, and comparative typology across known issues. When an image appears anomalous, the first question should be about the process that produced it. Who engraved the die, for what audience, and under what constraints? Only then can we consider biological explanations. Still, even when we account for artistic process, some images carry specific marks that invite interpretation. A child's bust showing asymmetry, or dental irregularity preserved on bone would be stronger evidence than a stylized coin. That brings us to the domain of osteology, the study of bones. Human skeletons preserve certain markers of congenital conditions: cleft palate scars in the maxilla, craniosynostosis visible in skull sutures, or anomalies in vertebrae and long bones. If royal remains preserved such indicators, and if those remains could be securely linked to named offspring of Cleopatra, then a case could be built. But again, the archaeological record is cold on that point.
Alexandria's funerary record is fragmentary. Royal tombs were targets for tomb robbers, and many ostensibly royal objects come to us through antiquarian channels that complicate attribution. The osteological route remains tantalizing but blocked by practical realities, indefinable. If osteology and ADNA are blocked, what remains are texts and the interpretive apparatus around them. Ancient descriptions of the Ptolemaic family, filtered through Roman writers with clear agendas, must be handled with care. Some passages emphasize the extraordinary, elaborate banquets, theatrical gestures, and displays of exoticism. In the Roman imagination, foreignness becomes a weapon. Children of a queen who challenged Rome could be portrayed as uncanny, not because they were objectively strange, but because their existence made Rome uncomfortable. Rumor may have amplified physical difference as moral difference. Part of the rhetorical strategy of Roman historians was to make the foreigner's body stand in for a political threat. Yet readers of antiquity sometimes mistake rhetorical amplification for eyewitness anatomy. That is a core danger for our modern inquiry. Sensational claims about appearance can leap from propagandistic text into modern scientific-sounding narratives because they satisfy a certain appetite for origin stories, the idea that political hubris or dynastic arrogance produced a literal bodily price. The thriller-like tagline, genetic revenge, plays into that appetite by personifying heredity as a moral force that punishes. But genetics is amoral arithmetic. It does not moralize. To marry the rhetorical punch of ancient slander with the precise language of modern genetics is to risk a category error. So where does that leave us at this stage of the investigation? We have established the context, a dynasty that practiced close-kin marriage, surviving visual records that are stylized and often ambiguous, textual sources that are rhetorically charged, and a scientific toolkit that promises answers only if it is allowed to operate within its limits. The next move is to interrogate the visual archive in fresh detail and to provide a clear, accessible primer on the genetics involved. Only with that groundwork can we test which hypotheses survive scrutiny. We will proceed by cataloging the key visual pieces associated with Cleopatra's household, assessing their provenance and the degree to which they might reflect anatomy versus artifice. We will then elaborate the basic genetic models that explain how consanguinity raises risk without guaranteeing phenotype, using simple family tree examples and cautious probability language. After that, we will examine what aDNA research in Egypt has actually achieved in recent years, where it has succeeded, where it has stumbled, and how that bears on the possibility of extracting data from Ptolemaic remains.
At the end of that diagnostic sweep, we will be ready to compare explanatory weight. How well do genetic mechanisms explain the available evidence compared to iconographic convention, political smear, or environmental illness? We are not traveling to a predetermined verdict. We are assembling the pieces so that each claim can be tested against them. Evidence that is missing must be named, probabilities must be stated clearly, and the human lives at the center of the story must not be reduced to laboratory samples or rhetorical devices. Now, let us examine the coins and sculptures themselves. What do their details reveal when we hold them up to careful scrutiny? What can the dye work tell us about workshop practices? Which portraits were state issues and which were private commissions? Where do patterns repeat and where do they break? In this next section, we will conduct close visual readings of a representative set of coins, matched with numismatic scholarship and technical analysis. We will treat each image as a source with its own biases, not as a literal mirror. As we approach the visual dossier, keep in mind the central caution: seeing is not the same as knowing. An unusual jawline on a coin might be an artist's shortcut. An asymmetrical skull might be a later restoration. A rumor might be a political weapon disguised as observation. Only by weighing all these elements together can we begin to separate the plausible from the provable. We begin with the coins, because they are the most numerous, the most mobile, and the most intentionally public images the Ptolemies ever produced. A coin carried the ruler's face into markets, into temples, and across borders. It was propaganda that fit in a palm. That means every technical choice, from the depth of the die, to the scale of the relief, to the arrangement of hair locks, mattered. It also means that coins are both invaluable and treacherous as biological evidence. They tell us what a state wanted to show, not necessarily what a child actually looked like. Numismatists read dies the way detectives read fingerprints. A die cutter's habits leave telltale marks: the way curls are carved, the angle of an eyelid, the particular groove used to render a chin. When the same die is used over decades, or when a workshop maintains a stable repertory, faces repeat. That repetition is a house style. For the Ptolemaic mint in Alexandria, a handful of facial templates survive across multiple issues. These templates often emphasize certain features: a pronounced brow, a firm mouth, heavy hair treatments, because they signal authority, continuity, or ethnic hybridity to diverse audiences. A repeated brow line does not equal a repeated jawline in the DNA. Technical constraints also exaggerate features. Dies were tiny sculptures. To survive repeated striking, details had to be bold. Shallow relief tends to flatten nuance. Deep carving throws shadows that sharpen lines. When a portrait is rendered in small metal, a small asymmetry on a living face can become a conspicuous trait. That is a mechanical distortion, not medical evidence. Provenance complicates matters further. Many coins we see in collections passed through centuries of circulation, hoarding, and loss. The sample is not random. Coins that survive tend to be those hoarded in pockets, hidden in deposits, or rescued from shipwrecks, not an unbiased snapshot of face types as worn and seen by contemporaries. Moreover, die attribution is a specialist discipline. The same visible quirks that make a coin look odd to a non-expert may be a marker of a particular workshop or an economy of production. Turning to sculptural evidence, the terrain grows murkier. Marble and bronze offer the potential for three-dimensional realism, but the survival path of these objects is littered with complicating factors. Many sculptures we associate with the Ptolemies were moved, repurposed, or restored in later periods. A head discovered in a Roman villa could be a commissioned homage rather than a royal portrait. Architectural fragments recycled in later constructions can be misread. Restoration efforts in the 18th and 19th centuries sometimes replaced missing pieces to satisfy collectors' tastes, producing faces that hybridize ancient workmanship with modern imagination. Even when an object is datable to the Ptolemaic period, distinguishing a royal commission from a workshop copy can be difficult. When a portrait shows an anatomical asymmetry that survives into the marble, that is interesting in a way a coin cannot be. Asymmetry, cranial features, and dental traits are the kinds of things that could, in principle, be matched to osteological markers. But the complicating reality is that many of the sculpted oddities turn out to be stylistic. In Hellenistic art, individuality coexisted with type. A sculptor might model a face with a hard lower jaw to suggest determination or to mark the subject as of a particular lineage. The criterion for interpreting such a trait as biological rather than symbolic must be strict: repeated appearance in securely dated, securely attributed royal commissions, corroboration from other media, and ideally osteological confirmation. Which brings us to what bones could tell us. The skeleton stores a record of some congenital conditions. Certain craniofacial anomalies, dental malformations, skeletal dysplasias, and signs of developmental stress leave marks on bone. A cleft palate can leave telltale traces in the maxilla. Craniosynostosis alters sutural patterns. Some syndromic conditions produce characteristic facial or limb anomalies visible in bones. In the best-case scenario, osteology and aDNA can corroborate each other. A bone abnormality that aligns with a pathogenic variant found in ancient DNA is strong evidence of a hereditary condition. But for the Ptolemies, that chain breaks.
Alexandria, the political heart of the late Ptolemaic state, is also the Achilles heel of archaeological recovery. The ancient city sits at a complex, waterlogged coastal environment. Modern urban layers cover and disrupt ancient deposits. Many elite burials from the late Hellenistic period were looted in antiquity. The few burials that survive are often fragmentary and hard to attribute to a named individual. When archaeologists have proposed royal burial candidates, their arguments rely on combinations of grave goods, architecture, and inscriptions, but even well-argued attributions are debated. In short, the chain that would connect a particular skeleton to, say, Caesarean, is not currently secure. That unknown, indefinito, is the central empirical barrier. Ancient DNA adds another layer of complication. The field of paleogenomics has made dramatic progress in favorable contexts. High-latitude, arid, and cold environments preserve DNA relatively well, but the Eastern Mediterranean is a middle ground. Humidity, fluctuating temperatures, and microbial activity accelerate DNA degradation. Nonetheless, aDNA work in Egypt has advanced. Researchers have recovered genomes from some Egyptian mummies and from Nile Valley sites. But the bulk of that work to date has not targeted individuals who can be confidently assigned to Ptolemaic royal burials. Most samples come from commoner contexts or from populations outside Alexandria's elite necropolis, indefinito. Where DNA has been retrieved, the standard tools would allow geneticists to estimate levels of homozygosity, to detect runs of homozygosity that imply recent inbreeding, and to screen for known pathogenic variants. Yet even with a clean genome, linking genotype to phenotype, particularly for complex facial morphology, remains hard. Let us pause in the workshop and outline in clear terms what genetic evidence would look like. When we talk about inbreeding and its possible phenotypic consequences, several technical concepts matter. First, the inbreeding coefficient. This is a measure of how likely it is that an individual inherits the same allele from both parents because those alleles came from a common ancestor. The closer the relationship of the parents, the higher the coefficient. A child of full siblings has a higher expected coefficient than a child of first cousins, and so on. Over many generations of close kin unions, the overall genomic homozygosity of lineage can rise. Second, runs of homozygosity, ROH. These are long stretches of DNA where both copies are identical because they descend from the same ancestral chromosome. Long ROH are signatures of recent inbreeding.
Many short ROH scattered across the genome can indicate more distant population history or small population size. Geneticists can calculate ROH lengths and infer whether an individual's parents were closely related. Third, pathogenic variants.
Some genetic disorders are caused by single gene mutations, Mendelian conditions, that are recessive. If both parents carry a deleterious recessive variant, their child has a measurable chance of expressing the disease. Examples in modern medicine include cystic fibrosis or Tay-Sachs disease in certain populations. If such a variant were present and homozygous in an ancient genome, it could plausibly be linked to clinical features visible on skeleton or in historical accounts. Fourth, polygenic traits. Complex aspects of facial structure arise from the interaction of many genes plus environment. Genome-wide association studies, GWAS, in modern populations can associate certain alleles with facial dimensions, but those associations are probabilistic and population-specific. Predicting a full face from ancient DNA is beyond present capability. You can speak to tendencies and risk scores, not to a portrait. Fifth, founder effects and drift. When a small subset of individuals gives rise to a population, rare variants can become relatively common. The Ptolemaic dynasty began with a small Macedonian elite intermarrying and ruling over Egypt. Founder effects could, in theory, concentrate certain alleles within that ruling house, but detecting such a signal requires comparative genomes from both the dynasty and the broader population. In practice, then, a confident genetic case for recurrent congenital traits in the Ptolemaic line would require 1. Authenticated skeletal remains assigned to named royals, 2. Endogenous, uncontaminated ancient DNA sequences from those remains, 3. Evidence of long runs of homozygosity or identified pathogenic variants consistent with the skeletal anomalies, and 4. Congruent osteological signs. Absent those convergences, the genetic argument remains an informed possibility, not a settled fact. Now consider how historians, numismatists, and geneticists might weigh the available evidence against alternative explanations. The alternatives are many and plausible. One major class of alternatives is artistic convention. Hellenistic portraiture operates with types and signs. A heavy chin may signal virility or authority. A square brow may be the signature of a mint master. A child's portrait rendered with adult contours may be an attempt to portray dynastic succession rather than juvenile features. These are not frivolous counterarguments. Portrait conventions explain a great deal of the repetitive appearance of Ptolemaic coin faces and sculptures. A second class is political slander. Ancient writers, especially Roman ones, had reasons to depict Cleopatra and her family as foreign, lascivious, or decadent. Describing heirs as physically odd served rhetorical purposes. It was a means of portraying political threat as moral and physical otherness. That rhetorical machinery is well documented elsewhere in antiquity. Bodies become symbolic shorthand for politics and culture. Thus, a Roman claim that a child looked strange tells us more reliably about Roman anxieties than about cranial indices. A third class is environmental and medical. Disease, malnutrition, infections, or developmental conditions unrelated to heredity can produce physical features that contemporaries notice. Childhood illnesses that affect dental development, cranial growth, or facial symmetry can leave marks on bone and impressions in narrative memory. The Ptolemaic period was not free of endemic disease. Provisioning, urban sanitation, and childhood mortality all shaped bodies. If a series of children in a royal household experienced similar environmental stressors, diet, infectious disease, or in utero insults, their appearance might have clustered in ways that created rumor without implying shared genetic pathology. A fourth class is selection and survivorship biases in the sources. Texts that survive are not neutral. Images that survive are not representative. If hostile sources wrote more energetically about the weird or the scandalous, and if collectors disproportionately preserved certain coins or statues, our modern sample will skew toward the sensational. That skew feeds the modern appetite for dramatic explanations. None of these alternatives eliminates the possibility that heredity played a role. In fact, they may operate in combination. A genetic predisposition made more likely by consanguinity could be amplified by environmental stress and then exaggerated in writing and in art. The more difficult but honest move is to model those combined effects probabilistically and to acknowledge the limits of inference. Comparative history helps, but must be used cautiously. The Habsburg jaw is a familiar example of a dynastic phenotype associated with long-term intermarriage. Portraits, abundant records, and reasonably attributed remains make it a strong comparative case. In other dynasties, however, similar rhetorical amplification has produced mythical physical traits with little anatomical basis. For the Ptolemies, the comparative literature suggests that long-term consanguinity increases risk but does not guarantee a particular syndrome. Context matters. Population genetics of the founding pool, the degree of outmarriage, demographic bottlenecks, and environmental pressures all shape the outcome.
Given this complexity, historians and scientists take different but complementary pathways to the same question. Historians parse texts for motive, bias, and rhetorical context. Archaeologists evaluate material context and provenance. Numismatics map dye groups and stylistic networks. Geneticists look for genomic signatures that are robust to contamination and degradation. When these disciplines converge, they can speak with uncommon confidence. When they diverge, the honest answer is uncertainty. Where could future research make the difference? Several realistic possibilities exist, though each faces practical barriers. Archaeology.
The discovery of a securely attributed royal burial, excavated under modern protocols with clear context in inscriptions that tie the remains to named individuals, would be a game changer. Alexandria's urban complexity makes such finds rare, but not impossible. Ongoing work in Egyptian necropolies, combined with improved remote sensing and targeted excavation, could turn up relevant contexts. Ancient DNA. Advances in extraction methods, library preparation, and computational decontamination are increasing success rates in challenging environments. Methods that sequence ultra-short fragments, target mitochondrial DNA as a first screen, or enrich for particular genomic regions can make retrieval more feasible. However, the success of aDNA depends on preservation conditions that are, in the case of Alexandria, often unfavorable. Even so, a carefully excavated, well-preserved sample could produce data sufficient to estimate ROH and detect certain pathogenic variants. Osteology and paleopathology. High-resolution imaging, micro-CT, 3D surface scanning, and paleodental analysis can detect subtle congenital markers invisible to the naked eye. Combined with isotope analysis for diet and trace element exposure, osteological study can differentiate between inherited developmental anomalies and environmental causes such as malnutrition. But that work depends on access to securely attributed skeletal material. Interdisciplinary integration. The real progress would come from converging lines, a find with good archaeological context, clean aDNA, detailed osteological analysis, and careful reading of contemporary images and texts. When those threads come together, the probability landscape sharpens. Lacking them, the landscape remains foggy. Beyond the must-have data, there are also ethical considerations that should guide research and presentation. First, the language used to describe potential findings must be precise and deferential. The phrase, "never looked human" in our title is deliberately provocative. It names the sensational claim so we can disarm it. But if future research uncovered a congenital syndrome in a royal line, describing living or dead people as less than fully human would be morally abhorrent and historically misleading. The aim of scientific and historical description should be to explain, not to stigmatize. Second, researchers must avoid presentist moralizing about appearance. Descriptions of physical difference in ancient sources often served political ends. Repeating those descriptions uncritically perpetuates the same rhetorical violence those sources practiced. Responsible scholarship contextualizes rhetorical motives and reframes physical descriptions in terms of evidence and probability. Third, scholars should be transparent about limits. The public is hungry for definitive narratives. Scholars must resist the temptation to offer tidy answers when the evidence supports only degrees of likelihood.
Clear caveats, accessible explanation of methods, and open sharing of data and reasoning are the best antidotes to sensationalism. With these constraints and possibilities in view, how do we synthesize the evidence? We can, at minimum, assemble a careful hierarchy of claims. Claim A. The Ptolemaic dynasty practiced close kin marriage, which increased the population-level risk of recessive disorders in their descendants. This claim is well supported by historical records of marriage practices and by general principles of population genetics. It is a plausible, even probable mechanism for increased risk. Claim B. Some ancient texts and visual depictions portray Ptolemaic children and royals in ways that later readers have described as unusual. This claim is supported by surviving sources. However, those portrayals must be interpreted within the rhetorical frameworks that produced them. Claim C. The existing archaeological and genetic record does not provide definitive proof that Cleopatra's children bore hereditary conditions that made them look markedly different from contemporary norms. This claim is supported by the absence of securely attributed royal remains with corroborating aDNA and osteological data, indefinito. Claim D. It is possible, though not proven, that a combination of hereditary risk, environmental stressors, and political caricature produced a persistent impression of physical oddity around the Ptolemaic children. This synthesis is plausible but probabilistic. It is the best fit explanation given current evidence. That hierarchy lets us answer the title question responsibly. The phrase, "the genetic revenge," suggests a moralized narrative in which heredity enacts a dramatic punishment on a family for its choices. Biology does not moralize, it changes probabilities. The best evidence-based response is that heredity could have contributed to physical features in Ptolemaic offspring, because the social structure of the dynasty increased genetic risk. But there is no conclusive evidence that Cleopatra's children never looked human, and there is no clean line from the documented marriage practices to a specific syndrome or set of facial traits. The accusation, or the chilling story, has a plausible mechanism, and it draws on ambiguous textual and visual traces, but it lacks the hard convergence of bones and genomes necessary for certainty. This verdict has broader implications. It teaches a method more than it settles a sensational thesis. It shows how three kinds of sources, texts, images, and biology, must be read together, and how each can mislead if isolated. It demonstrates why sensational narratives about bodies in the past are attractive. They bind moral drama to visual evidence, and speak to contemporary fears about heredity and identity. And it reminds us that historical inquiry risks repeating the same rhetorical harm of ancient slander, as long as it remains attentive to context and ethics.
What then would a responsible next step for researchers and for public communicators be? For scholars, investment in interdisciplinary projects that combine excavation in candidate sites, rigorous conservation and chain of custody protocols for recovered material, and advanced aDNA methods tailored to challenging preservation conditions should be prioritized. For curators and numismatists, continuing to publish high-quality dye studies and to digitize collections with provenance metadata will improve the sample and reduce selection bias. For communicators, the obligation is to present probabilistic findings with clear qualifiers and to avoid repeating rhetorical tropes that dehumanize. For the public, the lesson is twofold. 1. Appetite for dramatic stories about bodies and bloodlines must be tempered by an understanding of how evidence operates, what it can show, what it cannot, and why gaps matter. 2. Curiosity about the past is valuable when it is disciplined by method and by respect for the people who, though long dead, are still the subjects of narratives that shape how we understand ourselves. We close where we opened, with an image. Picture a small, well-struck tetradrachm, its silver rim nicked by circulation, once slipped into a merchant's purse and later recovered in a hoard. On one side, a familiar face and profile, the hair worked into ordered locks, the brow angled with the economy of the die cutter's tool. The coin carried authority, it repeated a type, it told a story the state wanted told. Now imagine that coin in a modern hand, photographed and uploaded to social media with a sensational caption. The coin travels again, carrying with it centuries of interpretation, invention, and selective survival. Our work as investigators is to return that image to the context in which it was made, to test its claims against what bones and genes and careful reading can tell us, and to report what we find without turning people into metaphors. The Ptolemaic story resists tidy endings precisely because the evidence resists closure. Cleopatra's children lived at the intersection of empire, representation, and rumor. Their faces were political, their lives were bound to forces beyond their control. To say that heredity may have increased their biological risk is to say nothing about their worth as people. To say that current evidence does not prove a dramatic genetic revenge is to insist on a standard of proof appropriate to both science and humane history. If future discoveries, a securely identified tomb, a clean aDNA sequence, osteological signs that match genetic findings, alter the balance of probabilities, the conclusion must change. Science advances by updating claims in light of new data. Until that day, the most defensible position is cautious curiosity, recognizing the plausibility of genetic risk without succumbing to the siren song of a neatly moralized bodily parable.
If this investigation has done its job, it will have shown why certain stories about the past are so persistent, and why the past sometimes seems to answer questions we are asking today. The Ptolemaic children continue to provoke because they stand at the juncture of power and image, biology and rumor. Our responsibility is to approach that juncture with tools that are sharp, methods that are humble, and words that do not amplify the very violences we claim to diagnose. For further reading and detailed notes on the sources that inform this investigation, consult the companion bibliography linked below. There, you will find suggested numismatic catalogs, collections of classical sources with critical editions, and accessible introductions to paleogenetics tailored for historical inquiry. Those resources will also indicate where the record is silent, the indefinidos, and what specific discoveries would be needed to resolve them. Thank you for joining this careful look at a provocative question. If this inquiry sharpened your thinking about how history, art, and science intersect, follow the companion notes for source references, and stay tuned for our next deep dive into the visual politics of another ancient dynasty.